Good morning, all.
Morning, ma'am.
Good morning. Studying? Are you all studying?
Yes, ma'am.
Don't waste your time. You have to start studying. Okay. If you don't have books, I think you have your PDFs. See, the attendance is so low. Like only some of you are attending the classes and some are missing. Many of you are missing the classes. I don't know the reason for this. Today I will take the demo of the organ testis. Is it visible?
Yes, ma'am.
Oh.
Yes, ma'am.
Okay. So, theory of this is over already, right? So, only the demo of the organ I will be taking. The testis belongs to male genital organ. In the male genital system, we have the testis, then the epididymis, vast difference, then the prostate gland and the seminal vesicles. The bulbourethral glands and prostate gland will help in increasing the secretion of the fluid, that is the semen. They add to the products of the semen. Now each testis is hung in the scrotum with the help of the spermatic cord. This is the spermatic cord. This is spermatic cord. Here, the spermatic cord has got 3 coverings. It has 3 coverings from outside to inside. It is covered by external spermatic fascia. I think I will not be so clear with these fascias. So as much as I can show, I will demonstrate here. It is covered by external spermatic fascia. Then there is a cremasteric fascia and internal spermatic fascias. 3 coverings will be there, covering the spermatic cord. Now you should know the derivation from which these layers are derived. That means the external spermatic fascia is derived from, can anyone answer?
External oblique muscle.
Yes, external oblique muscle. Then you have the internal oblique muscle will give rise to the cremasteric fascia and also few fibers, muscle fibers. Then there will be the internal spermatic fascia. So all the coverings are here in this. Here, they are all in this. Then internal spermatic fascia is derived from the fascia transversalis. Here, all the 3 coverings will be there. Now, when we come to the testis, I will stand up, this will be better. Okay. So here, the right and left testis are hung in the scrotum. The left testis will be at a slightly lower level than the right. Clear? Left testis is slightly lower than the right. Now, presenting features of the testis. It has a upper end. I'll take the other one. Okay. So this is the upper end of the testis, upper end. Then a lower end. Then there will be anterior border here. This is the anterior border. Then a posterior border. Then a medial surface. A medial surface and a lateral surface. Here, so presenting features of the testis are the upper end of the testis. This is the upper end, the lower end of the testis. Then you have an anterior border. Here, this is the anterior border. Then a posterior border, the medial surface, and a lateral surface. Here. So upper end of the testis will be covered by, you can see here, it will be covered by the head of the epididymis. Can you see here? This is the head of the epididymis. So almost it will be covered by this. Then lower end will be occupied by the lower end will be occupied by the tail of the epididymis and the beginning of the vas deferens. See here. So this is the lower end of the testis, which will be occupied by tail of the epididymis and also continuation of the vas deferens. So as much as you can appreciate, just to try to appreciate these here, tail of the epididymis and continuation of vas deferens. Then the anterior border, this is the anterior border. Anterior border will be, it will be smooth and convex. Here, anterior border of the testis is smooth and convex, whereas posterior border will be, it will be broad. This will be broad. Here, posterior border is broad. Then medial surface is, see this is the medial surface. This is also smooth. Whereas lateral surface, you can identify on the lateral surface posteriorly, there is a small sinus gap is there. Can you make out this gap? A gap is there between the testis and the epididymis. So this gap here, this gap is called as sinus of epididymis. Here, sinus of the epididymis. Can you make out this gap? So this gap is present on the posterolateral part of the testis. So if I hold it like this, here anterior border, posterior border, and laterally near the posterior end, laterally, posteriorly, there is a sinus. So this sinus will help in determination of the side of the testis. Otherwise, you cannot identify which side it is. Clear? So you have to make out this sinus, presence of this sinus, which helps you to identify the side of the testis. Is it clear? So this one is, so if this is lateral, that means it is left side. Left side. Now coming to the coverings of the testis. So there are external coverings and extrinsic coverings and the intrinsic coverings. So the intrinsic coverings they belong only to the testis, whereas the extrinsic will cover the wall of the scrotum also. Coming to intrinsic coverings of the testis. So one covering which I can show you here is the tunica vaginalis. This one. Can you see here this one? This is tunica vaginalis. So even in tunica vaginalis, there are two layers, the parietal layer and a visceral layer. Only the visceral layer belongs to intrinsic covering. Clear? Whereas parietal layer will also cover the scrotum, so it becomes extrinsic covering. So here, this is the visceral layer of the tunica vaginalis. Then one covering will be there which is very much adherent to the testis. You cannot separate it from the testis. That is tunica albuginea. Clear? Tunica albuginea. Now this tunica albuginea, it enters inside the substance of the testis and it, see here, this is the cut section here. Clear? So tunica albuginea will enter inside the substance of the testis and divide the testis into small lobules. There are about 200 to 300 lobules inside the testis. Here, so tunica albuginea enters inside and it covers every lobule. So that lobule will be a conical shaped structure. So if you can appreciate from here that conical shaped lobule, it's good. Okay. Can you see here conical shaped structures? Here, they are all the lobules of the testis. Each one is covered by tunica albuginea. The next covering is tunica vasculosa. Here, so this tunica vasculosa will be covering inside one lobule. So inside each lobule, there will be a coiled structure called as seminiferous tubule. So these, see, minute, can you make out in this? I'm not sure whether how much you can appreciate. These minute coiled structures here, what are seen here, these are all the seminiferous tubules. Here, every seminiferous tubule is covered by tunica vasculosa. Here, tunica vasculosa. So, three coverings are there. Tunica albuginea, that is the parietal visceral layer. Then you will have the tunica albuginea, and lastly the tunica vasculosa. Now, what are the extrinsic coverings? So extrinsic coverings, I told you, they will also cover the wall of the scrotum. So you will have the parietal layer of the tunica albuginea. Okay. So if this is your visceral layer here, this is the visceral layer. This is the visceral layer here. There will be another layer. Both of them will become continuous at the posterior border of the testis. So here they will create one sac. So it's just like how you have a pleura or a pericardium. Like that, there are two layers in the tunica albuginea. Both of them form a sac here. So that sac usually, it does not contain any fluid or anything. That is the processus vaginalis. And further, during the descent of testis, this helps, processus vaginalis brings down the testis along with the help of the gubernaculum testis. Yeah. So usually there is no fluid between the visceral layer and the parietal layer. But sometimes maybe a congenital cause may be there. Okay. What happens will be like you'll have a fluid collection between the two layers here. that will lead to hydrocele. Or sometimes you see that the processus vaginalis persists, carrying contents of the abdomen, congenital variation I'm telling here, and it may lead to congenital hernia. Here, congenital hernia, which is extending throughout into the from the inguinal canal through the spermatic cord and to the up to the base of the scrotum, and also the from the apex up to the base of the scrotum. So that is a congenital hernia if it persists. Here, carrying the abdominal contents. Or if fluid collection is there, it will be called as hydrocele. Okay. That is about coverings of the testis. Now, at the upper pole, the upper pole of the testis contains a small sessile pedunculated vestigial structure. Can you make out this? Small pedunculated, a small dot-like structure. Can you make out here? At the upper end of the testis, here. See, this is pointing here. Okay, this is the remnant of the Mullerian duct. Remnant of the Mullerian duct. Appendix of the testis. This is called appendix of the testis. See this properly. Small one, a small structure here at the upper pole of the testis. Okay. Now, see the cut section of the testis. Right. Now here, from the anterior end, the tunica albuginea, that will enter inside and divide this into number of 200 to 300 lobules. Clear? Now, each lobule will be containing the coiled seminiferous tubules. Now, all these seminiferous tubules, they will end up in efferent ductules. So here you can see these are the efferent ductules. Clear? They are the efferent ductules. They form from the rete testis. So I think this is all not appreciable in this. Okay. So a network will be formed. From there, the efferent ductules will join the head of the epididymis. So what is this head of the epididymis made up of? Head of the epididymis, here you can see, head of epididymis is containing the coiled efferent ductules will be there. Clear? The whole thing will form one duct, and then in the body of the epididymis will be containing just a single duct of the epididymis. And then the epididymis continues as, see here, this is the tail of the epididymis, continues as the vas deferens. So this structure is the vas deferens. This vas deferens enters inside, this is the vas deferens, enters inside the spermatic cord. The length of the vas deferens is about 45 centimeters. It is 45 centimeters. So that is about the features of the testis. Now I told you about the spermatic cord, right, coverings. What are the contents in the spermatic cord? So if I can just show you here, all the contents are dissected here. Here. So you have to identify these structures in the spermatic cord. So one which are looking bluish, can you appreciate that bluish? Okay, these are nothing but pampiniform plexus of veins. Pampiniform plexus of veins, testicular veins. You have heard of testicular veins, pampiniform plexus of veins, right? These bluish structures are pampiniform plexus of veins. Then, how to identify other structures? See, one thing we can identify here is a cord-like structure within the spermatic cord, that is, okay, I am feeling here a cord-like structure. Here, this cord-like structure is vas deferens. That is vas deferens. Now, how to identify why it is cord-like? Can any one of you tell why it is cord-like? Vas deferens is cord-like, very thick. Yes, answer me. Vas deferens is thick and cord-like. Why it is so? Am I audible?
Yes, ma'am.
Yes,
Yes, ma'am.
You answer them. Why vas deferens is cord-like? See here, this I can feel it. That's why I am able to identify that. Okay. So why it is cord-like? Why do you think it will be cord-like and very thick? What makes it cord-like? Anything which is very firm means what will be there? Okay, I separated it also. See this one. This is vas deferens, this structure. The musculature in the vas deferens is very thick. Okay, so that is the reason the circular muscle coat of the vas deferens is very thick. It is even thicker than the lumen of the vas deferens. Here, thicker than the lumen of the vas deferens. Why so? Because the muscular contraction of the vas deferens will help in transportation of the spermatozoa. So spermatozoa are stored in this epididymis, right? They should be transported against gravity, right? It should be transported against gravity into the inguinal canal and then into the pelvis, right? So if it is against gravity, the motility of the sperms will not help here. Here, it won't help. So you need some extra action for that. That is contributed by the thick muscular wall of the vas deferens. The contraction of this muscle coat of vas deferens helps in transportation of the spermatozoa, clear, against gravity. Okay. Then, what are the other structures in the spermatic cord? You have many arteries. So, artery to the vas deferens. Here we can't identify everything here. Okay, the whole bunch contains artery to the vas deferens, artery to the testis. Okay, testicular artery. Then you will have cremasteric artery. The cremaster muscle is there, right? So that also should be supplied by artery. Then you will have the genital branch of the genitofemoral nerve. It's a cutaneous one, genital branch of genitofemoral nerve. Then you will have the lymphatics. Clear? All these are the contents of the spermatic cord. So this much is there for demonstration. So you will have many questions like the coverings of the spermatic cord, the contents of the spermatic cord. Clear? So read them up, draw the diagrams, learn the diagrams. You have to draw even the cross section of the contents and coverings of spermatic cord. Then in the testis, this whole organ will be kept for dissection. This one exam here, practical exam, and then histology part is important in testis. I will just show you the first one I showed was this testis, right? I determined the side. Now one of you can determine the side of this testis. I will just show you the features here. Okay. So here, this is anterior border. Okay, anterior border. Then here you have the posterior border. Then this is lateral surface. This is the medial surface. Here is the sinus of the, see here, sinus of epididymis. Identify the side. Yes, can you identify the side? This is
Posterior.
which side testis is it? This is anterior border. Okay. So I told you the sinus of epididymis will be present posterolaterally. So I am holding it posterolaterally. Which side is it now?
Right side.
Posterolateral. Yes, very
Right
good.
side.
It is right side. Okay. So that is how you have to identify the side. Clear? And did I show that remnant of the testis? I showed one. Is it appreciable, this one? Remnant of the Mullerian duct. This
Yes, ma'am.
one. You see this small one. And sometimes even over the epididymis also you'll find one small. That will be the remnant from the mesonephros. Okay. On this specimen, I don't find any small remnant over the epididymis. Here, some small fibrofatty tissue will be there here. That will be remnant of the mesonephros. Okay. So once you come back again, we'll give these organs for revision. Don't worry. But however, you have to start reading. If you don't have your books, start reading from the PDFs. Already online free PDFs are there for you, right? You have any doubts in this? You can ask. Do you have doubts?
No, ma'am. Ma'am,
Yes.
will you please tell again why ductus difference is cord-like?
Because the this one, that is the epididymis, will store the sperms, right? Spermatozoa. That has to travel through the vas deferens, because vas deferens begins from the epididymis, head of the tail of the epididymis, right? So here is the, you can see here, this is the, yes, this is head of the epididymis, and then the tail will be here down. Then begins the vas deferens below. Here. So this vas deferens is about 45 centimeters. Here, thus that vas deferens has to travel all along the spermatic cord, right? Then it has to reach through the inguinal canal. Then from the inguinal canal through the deep ring, it will enter into the pelvis. Then it will drain into the along with the duct of the prostate. It will become the ejaculatory duct and drain into the prostatic part of the urethra. Clear? So this much length will be there. 45 centimeters it is. 45 centimeters is not a small length, right? So the sperms have to travel against gravity. So that means something extra force is needed for the sperms to travel against the gravity. So if we, some extra force is provided by what? This musculature of the vas deferens. Here, the musculature, contraction of this musculature will help in transportation of the sperms.
Thank you, ma'am.
Okay. Any other doubts? See, the attendance is very low, only 79. So you have to motivate your friends. What else can we do now? See, only 79 of you are there. We are taking our whatever best efforts we can do. Motivate your friends to attend the class. Don't waste time. Okay, you can leave the meeting. Thank you.
Thank you, ma'am.
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 Thank you, ma'am.